Hi Year 12s, this is going to be our second video on the topic of the cell membrane. In this video we're going to be talking about how transport occurs across the membrane, so we're going to be looking at things like diffusion, active transport, endocytosis, exocytosis, things like that. To prevent any more time, let's continue. So first off, we're going to look at diffusion as osmosis. So we have two diagrams on those on the side. So we have diffusion on the left here, and we have osmosis on the right. Both of these processes are really the net movement. So if we start by looking at diffusion, diffusion is the net movement of particles from a region of high concentration to a region of low concentration. And that will keep going until they reach equilibrium. So there's the same concentration on both sides, or isotonic on both sides. Now, truthfully, that doesn't mean that's the end of diffusion. Those particles will continue to exchange one or two every now and then, because remember, they're just bumping into each other repeatedly. They're going to knock each other over that membrane. And in order to keep themselves at that isotonic level, diffusion continues to ensure that we remain at those levels. Now we say it's until equilibrium because at equilibrium there's zero net movement, which is why diffusion is the net passive movement. So it's just looking at the summary rather than the individual movement of particles. As we can see in the animated GIF over here, we have a high concentration starting on the left, and as the GIF starts to play, those start to move across the permeable membrane, semi-permeable membrane, until there's an isotonic levels or equal concentrations on both sides. Osmosis, on the other hand, another type of passive transport, is the net movement of water molecules from a region of low solute concentration to a region of high solute concentration, also sometimes referred to as the diffusion of water. So again, over here we can say we have a very low solute concentration over here. There's no sugar particles, and on the right we have a lot of sugar particles, and that water diffuses across, trying to lower the concentration of the sugar on this side. Again, this is a net movement, so we say until it reaches equilibrium, but truthfully there is molecules still moving back and forth across, but the net movement is zero once you reach equilibrium. As I've mentioned sort of throughout both processes, when two solutions have the same concentration as each other, they're called isotonic. When concentrations are different across permeable membranes, we will always have net movement until we reach that equilibrium. They'll generally be hypertonic, which means they have high concentration, in terms of osmosis, they'll have the most water. So hypertonic, where there's the least amount of water. Osmosis in tissues and organs. So osmosis, truthfully, is something that we need to think of a lot in the medical environment. So there are medical procedures where patients have to receive tissues or organ transplants from other patients. In order to prevent osmosis, so we need to keep those organs having the same concentration of all their important chemicals and molecules. Generally, those tissues and organs will be soaked in isotonic solutions while they wait for them to be transplanted. Similarly, dehydrated patients are often given IV fluids. Just thinking for a second, if those IV fluids were just water, then as it's pumped into the blood, it would increase the concentration of water in the blood. That would mean that the red blood cells would start to take up water through osmosis, and because they're constantly taking water in, trying to reach that equilibrium, eventually the red blood cells will burst and they'll die. Now, red blood cells, their entire function is to carry hemoglobin throughout the body. So if you don't have red blood cells, the patient could potentially die because they're not getting oxygen transported throughout their body. So keeping everything at those isotonic levels is very important for the body. Facilitated diffusion, so this is another type of passive transport. That means it doesn't require energy, and that again means that we're going to be moving from molecules in a high concentration to molecules in a low concentration. Facilitated diffusion is different from simple diffusion in the fact that we need channel proteins or transport proteins for it to happen. This is generally because we might be dealing with a hydrophilic molecule that can't move across the membrane, or it might just be too large to physically move across the membrane. So channel proteins have a hydrophilic pore in the membrane. That allows small polar molecules to diffuse in and out of the cell, allowing them to avoid that hydrophobic section of the cell membrane. So they're attracted by the hydrophilic nature on the inside of the cell, so they can move straight through that channel protein without being hindered. Channel proteins can be opened or closed. That allows us to help control the amount of particles that are in a cell. It also means that they can be very specific, so they'll only allow one ion, for example, to move through each channel. Transport proteins, on the other hand, can help substances like glucose and amino acid into the cell. These molecules are too big to move via simple diffusion, so they have specific binding sites in which they'll bind to a protein. Once they bind to that protein, the tertiary structure will change, allow that molecule to move into the cell across the membrane, and then the transport protein will return to its original shape. Now, with simple diffusion, the rate is determined by the concentration and how fast they need to move across a semi-permeable membrane. With facilitated diffusion, while we'd like to say the same thing, unfortunately the rate of facilitated diffusion is actually determined by the availability of channel and transport proteins to move those molecules across into the cell or out of the cell. Active transport. Active transport is when we're moving from areas of low concentration to high concentration, and because of that we're moving against the concentration gradient, which means we require energy to physically make that transport occur. Active transport will not occur without ATP. Remember, we looked at ATP last week, adenosine triphosphate. Proteins that are involved in active transport are referred to as carrier proteins, and the process is very similar to facilitated diffusion, except they need energy to happen. If we look at the diagram on the right here, originally we're starting with high to low, but once we reach equilibrium, the actual process continues to occur. As you'll see to start with, energy is required. Energy keeps coming up to the carrier protein, which changes shape, forcing these molecules inside of the cell despite the concentration on either side. So we're making sure that we transport all of these molecules over regardless of the concentration gradient. Exocytosis and endocytosis are two types of active transport. So both of these are processes in which we are using the fluid structure of the membrane, remembering that the fluid structure of the membrane means that phospholipid molecules can be added or removed or move around without physically tearing the membrane. And that's all exocytosis and endocytosis is. It's the movement of particles in and out of the cell, but using those parts of the cell membrane. So we're adding or removing parts to create vesicles and push or take molecules into the cell. Focusing on endocytosis first, endocytosis is a process where we're moving molecules into the cell. So endocytosis, into cell. The cell membrane starts by engulfing the material to be taken into the cell, and that's very similar to how unicellular organisms actually feed. Two types of endocytosis are phagocytosis and pinocytosis. Phagocytosis is almost like a cell eating, so it's taking in solid materials, where pinocytosis is like the cell drinking, where it's taking in fluids. Both of them are very different processes. So phagocytosis here, type of endocytosis, you can see we have a solid particle and the actual cell membrane starts to pinch off and actually surround the molecule before pulling it inside the cell. And that part of the cell that's engulfing the solid particle actually pinches off of the membrane, making those phospholipid molecules removed, where it's moved into the cell and will be digested. Whereas a fluid is a little bit different. We can't just have the membrane coming out and grabbing and pulling in. It actually starts to move inwards into the cell like this, and our fluid starts to fill in. At that point, the membrane will pinch off, and again, we'll have phospholipid molecules removed, creating a vesicle, pulling our fluids into the cell. Exocytosis is very similar to endocytosis. In fact, it's the opposite reaction almost. Exocytosis, just the bulk transport of materials outside of a cell. So the material to be moved outside of the cell is packaged into a vesicle inside the cell, like this. The vesicle then moves towards the cell membrane, where the vesicle becomes merge with the cell membrane, creating and adding those phospholipid molecules to the membrane. When it starts to merge completely, it will start to force those particles that we want outside of the cell into the extracellular environment.